Assalamu alaikum wa rahmatullah. Peace be upon you all, and welcome to another live after show on the Seeing God in Biology series with Dr. Fuz Rana. Fuz, how are you today?
I'm doing great, Ahmed. I'm blessed more than I deserve. So even my bad days are still pretty good days.
Thank God for that. So this has been quite an interesting episode about the Central Dogma. As funny as the name of the Central Dogma is, but it is what it is. And maybe it is for a reason that God let them call it this way, more or less. All right, so let me tell you a story and get your opinion about it until we have some questions. So it was a few years ago, I think it was like 4 or 5 years, and I was in Germany and having a dinner with a person who was in the IT field, but then the person told me that he's actually a biologist. So so I immediately asked if the person believed in God, and was not very sure. And I asked, okay, you're a biologist and you know about DNA, and do you accept that the genetic code is like a program? And the person said yes. And I said, okay, so you are also in IT and you know that a program needs a programmer? And he said yes. And then I said, so who is the programmer? And I think the answer was like undeniable, but it was like, still don't know or don't want to take that step, sort of. So let's start the question with this. In various conversations that I have had just this one peculiar discussion with 2 or 3 evolutionary biologists in one debate I was having, in the after show of a debate. And I was saying, okay, this is information. It is coding for proteins. And those proteins are like machines. So the information is essentially a program, because the protein is actually a program, because this is what a program does. It takes inputs, it does a processing, and it produces outputs. And this is exactly what a molecular machine does. It takes some input, it processes it, and it produces an output. So this is like a physical program. This is how. So information that codes for a protein is actually information that manifests into a processing system. So it is actually a program. And I was just struck with this argument. I learned later that this argument, some evolutionists and some naturalists are trying to popularize this argument, as ridiculous as it would sound. I'm not sure if you have even heard it before. It's so ridiculous I couldn't have thought of it myself. And they say, okay, so you're saying DNA or genetic material has information. What is the definition of information? And okay, so I didn't know where the question was going. So I said, well, information, according to information theory, is a measure of uncertainty. This is what information is. Mm-hmm. And as peculiar as the definition looks, but hard luck, guys. This is how we strange people in information field have definitions. So it goes like this. So if I tell you, if it's in the morning and I tell you the sun is shining, there is no information, because there is very small level of uncertainty that the sun did not come up today. Okay, mm-hmm. Because obviously since today is not doomsday, then the sun must have come in the morning. Okay, but if I tell you while we are in the morning, if I tell you the sun did not rise today, then the information content there is, the first example, the information is almost very close to 0%. And the second one, the information is very close to 100%, because it is very informative to me to know that the sun actually did not rise today. And then, so I say, so information is a measure of uncertainty, and the more improbable a certain arrangement of characters in this case is, the more the information content is. And once you detect information content beyond what we would call random, beyond what we would call noise, then we know that probably there is a mind behind it. And then, so I say, since the information in DNA or the codes in DNA manifest in arrangements that are way beyond random, then they have to be the work of a mind. And then the absurd argument now, after the strange question, the absurd argument is, but how do you quantify information? Because if you cannot quantify it, then you cannot claim that there is information. And actually, the argument was so absurd, I was not sure where it's heading, because my argument was, if you look at the beginning of life versus today, for example, and you look, for example, at a human eye versus no eye at all, for example, before the Cambrian, then you can notice that there is information that came into the system, for example, to code for a visual system. And then I noticed why this person was putting forward the question, because he says, how can you say that, for example, an eye spot in an algae has less or more information than a human eye? This was the argument, and it was ridiculous. So I say, are you telling me that you cannot recognize that you need much more information to code for a human eye versus an eye spot or a photo spot in an algae? And the person's would take it to as far as, no, since we cannot really quantify information, we cannot tell if an eye spot has more or less information than a human eye. And the argument went on, and I noticed that they were just arguing for the sake of argument, because how would you respond to something like that? If you have never heard it before, I'm not sure if you have.
To me, I think that maybe we may not be able to produce maybe a rigorous quantitation of the information content. Let's say in a biological system where or in an organism where the visual system is an eye spot versus, something like the camera eye of a vertebrate, right? But we could simply look at the complexity of the genetic apparatus needed to create those systems, and then to direct their operation, and semi-quantitatively recognize that there's clearly a much more complex genetic apparatus. And so therefore you would say that it's clear that there's more information in one system than in the other. Or another way to think about it is, in terms of algorithmic information. I'm not an information theorist, so I very well may be wading into a quagmire that I don't know how to get out of. But so I'll trust in you to help me. But the algorithmic measure of information is interesting, because it's what is the extent of the instructions you need to produce that system, right? And so something like if you just had a string of in DNA, let's say a string of adenines on a one of the strands, and the other strand would be a string of thymines. Well, the instructions are very simple. Just repeat X number of times. But when you look at the sequence for a gene, the algorithm that you need to specify that gene is essentially the gene itself, right? That the set of instructions is as extensive or as complex as theoretically possible in order to account for that gene. And so taking that concept, you could argue that the instructions that you need, in a sense, maybe it would be, the number of genes, but the set of instructions you would need to produce, a camera eye of vertebrates compared to an eye spot, again, qualitatively, or semi-quantitatively, is clearly much more extensive. And when you start talking about, the developmental process, that set of genetic instructions is dynamic through the course of growth and development. So genes are being turned on and turned off, and it's different combinations of genes that are expressed at different levels that are actually, so there's an additional dimension, a time-based dimension to the information as well. So, and clearly, again, the complexity of that, the time-based algorithm is far superior or far more extensive in the case of a vertebrate camera eye versus an eye spot. So, I think it's, yes, maybe we don't know how to precisely quantify it, because I don't know that we've been able to actually, because of the complexity of the system, be able to actually catalog the entire information content. But qualitatively, I think anybody who's reasonable would think there's clearly a difference in the amount of information, and that the information is vast, and and the information content is dynamically changing with time. It's not static information that's directing development.
Absolutely. Actually, I gave an answer that is comparable to that. So I would say that we can also quantify the amount of information by putting together a minimalistic set of instructions to describe both systems. And obviously, if we want to describe an eye spot versus a human eye, then describe it, a full description. Then describing how to build an eye spot will take much less operating instructions, or whether the bill of material or how to assemble that bill of material into a functioning system in the first case is much smaller than in the second case. So you will start, for example, by looking how many proteins are there and how you assemble them together, and then how those proteins would produce that one specific organ, and then how those organs or those subcomponents, all of them will have to go to be arranged together, which will be then a reflection of the developmental aspect, etc. And I think they didn't like the answer, and they said, well, there is no way we will be able to convince you, and hence we call it a day. So I said, okay, let's call it a day. And I'm always surprised with this attitude of putting what I would consider absurd questions, because when things are intuitively obvious, sometimes it doesn't make sense to ask the question. If somebody is telling me, for example, prove to me, for example, that this 5-page book has less information than this 500-page book. So I'm not sure how what would be a reasonable answer to that. But anyway, we have another question here. So the question is, what about non-functional proteins? So maybe because in our episode, we discussed a lot about the production of RNA from DNA and then proteins from RNA. And then his question is, what about non-functional proteins? I'm not sure if there are, what that means, but then he asks, what designer do when protein don't function? I think he means he's denying that there is a designer if there is a protein that does not function. Number one, is there something that is called a protein that has no function? And hence, what is your comment to the whole aspect matter?
I am not and I could be proven wrong here very easily, but one of the things that I find interesting when it comes to models for molecular evolution. So here when I say molecular evolution, I'm talking about something different than chemical evolution, though the terminology superficially seems very similar. Chemical evolution would be the process associated with abiogenesis, whereas molecular evolution would be the term in which you now have an intact genome and in a fully functioning cell, and it's how do you create new proteins from pre-existing proteins, and it's usually some kind of mechanism like maybe gene duplication followed by some kind of evolutionary process. And one of the things that if that was indeed the case, then you would expect to see within the proteome a significant fraction that would consist of what I would say non-functional proteins. That would be these proteins that are transitional intermediates that are being expressed, but are not really performing any kind of function, where that duplicated gene would be waiting for new mutations to eventually accrue so that what it's transcribing eventually would be of some kind of functional utility. And I just simply don't know of anybody that has ever cataloged what you might call non-functional proteins. People have argued that there are pseudogenes in genomes, which would be, presumably genes that have been duplicated, that have been then, that duplicated gene undergoes mutation, or maybe it's a unitary pseudogene where it's a gene that encodes for a protein product that isn't necessary, not that it's non-functional, but it's not necessary for the survival of the organism. So mutations there, would have minimal consequence. Or sometimes there are what are called processed pseudogenes, where the gene would be transcribed and then that messenger RNA is reverse transcribed to DNA that gets incorporated into the genome, and it's a process. It's called a processed pseudogene. But the problem is that the more that we're learning about pseudogenes, the more that we're learning that these pseudogenes are functional. And there's an idea called the competitive endogenous RNA hypothesis that essentially explains why functionality for both unitary and duplicated pseudogenes, and also explains why these have similarities to intact genes, but yet why it's important that they actually don't code a protein product, but their transcripts are actually playing a role in gene regulation. And one of the simplest expressions of this competitive endogenous RNA hypothesis has to do with duplicated pseudogenes, where we now are learning that once messenger RNA is produced, everybody used to think, and we're going to do an episode on this in the near future, one of the ways in which people understood gene regulation to take place was you prevented the RNA from being transcribed. If you don't need the protein, when we're now learning that in addition to that point of gene regulation, there's also a process in which the RNA, once it's produced, is actually broken down before it can be translated. And so in order to manage the regulation of gene regulation with respect to that mechanism, you have to produce essentially the duplicate. You have to transcribe the duplicated pseudogene, or now you have messenger RNA that's actually functioning as a decoy. And that if you and so that by that messenger RNA that's not coding a functional protein gets destroyed, it allows for greater expression of the intact gene, so that because that messenger RNA is much more likely to survive. And so it's a mechanism known as competitive inhibition in effect. But the point, but this decoy mechanism actually has has been demonstrated to have biological consequences. There's an anti-tumor gene known as P10 that's involved in tumor suppression, and people have shown that P10 has there's a P10 pseudogene, and in mice, if you actually eliminate the P10 pseudogene, the incidence of tumors it escalates or elevates in the mouse model.
Wow.
And that P10 pseudogene is functioning as a decoy. But and so that's one aspect of the competitive endogenous RNA hypothesis. There's actually a very complex family of mechanisms that are at work involving pseudogenes, and so that would be an example of a something that has been deemed to be non-functional and therefore doesn't reflect design, that later turns out to be functional as we learn more about the system.
Yeah, another question that I was actually somebody sent me. Actually, it was not sending. It was when I was doing the solo after show of episode 4, and the question was about gene duplications actually, and so his question was, how come those gene duplications just happen, and he's saying that there is all of those what was used to be called junk DNA, and it just keeps copying itself to transposable elements, etc. So what's your comment? His question started about whether gene duplication makes sense, and I just gave a thesis that if a gene gets duplicated and then gets bombarded randomly, it will essentially be junk, and its very persistence means that it's not junk, because otherwise natural selection should take it out. But then the question is, if what is the use of this same gene copying and copying and copying itself so many times in the genome? Do we have enough knowledge now about what's really happening with transposable elements, etc., or this is still an infant field?
Yeah. I think it's fair to say that it's a field really in its infancy, and we are, when we see certain features in the genome, we make assumptions as to how those features arose. So we see a duplicated gene, excuse me, we assume that gene duplication happened by some type of genetic mechanism that people, present models for how that duplication can take place. But most of the time, the inference is when we see duplicated, let's say a duplicated gene, we're just assuming that duplication is the result of a biochemical mechanism versus an intentional design. Nobody usually actually goes out and does the experimental work to demonstrate it was actually the product of duplication. There's just an assumption that this is the case, and so we.
You mean that nobody has really been looking, and they see that this has actually, now it's 1, now it's 2, now it's 3. They just find all 3, and then they make an inference that this is coming from duplication. Okay, right. Very interesting.
And
okay.
Even when you see differences in copy numbers, those differences in copy numbers actually have functional significance as well. So we are making assumptions that certain features in the genome are non-functional. We produce mechanisms for how those non-functional features might arise through some kind of biochemical or process or some kind of chemical or physical insult. And we just assume that this is the explanation. But people have actually not experimentally verified in many instances that mechanism actually is what produced that feature. It's just assumed, or we see, what look like, transposable elements, and we just assume that these are non-functional sequences. Nobody actually goes out and does the experiment to show unequivocally that it's non-functional. And to establish absence of function is not necessarily easy to do in a complex multicellular organism, because you also have to factor in development, or just because it's non-functional in one cell type doesn't mean it's not functional in another cell type. So people haven't actually done those experiments. It's just that the evolutionary paradigm is influencing the interpretation of a particular genetic feature. And thanks to things like the ENCODE project and other independent studies, we're learning that virtually every class of junk DNA is functional. But not only that, we're beginning to develop an understanding as to why, those non-functional sequences have to be the way that they are, right? So, for example, I've written a series of blog articles about recent discoveries about endogenous retroviruses, as well as endogenous viral elements in genomes having function. And it turns out that one of the major functional purposes for these, viral elements in the genome is that they actually are part of the innate immune system. They actually have they play an antiviral role. And the way they mechanistically exert their antiviral effect has to do with the similarity of those sequences to viral genomes. So, and again, it's not just a single mechanism, but there's a growing number of different mechanisms connected to these, endogenous viral elements and ERVs that, indicate they're playing a role in innate immunity through a variety of different means. But all of them, again, are predicated on the similarity of those sequences to viral sequences. So this is an example of, to the point now, there's just a mounting number of studies that are, taking as matter of fact that ERVs and EVEs are actually functional. This is not even disputed anymore, or not considered even an unusual result. But these discoveries never make their way into the kinds of conversations that we're having. People are just simply not aware of them. But the experts in the field, this is non-controversial, and so I guess the broader point is there's a lot of assumptions that are being made about the origin of these sequences and their functional utility that have not been actually experimentally verified, but are just assumed to be the case because this is what the evolutionary paradigm would demand, or would predict, or would expect, right? And yet, from a design perspective, we would say we expect these sequences to be functional, and we expect, that there's a reason why they look the way that they do, and those expectations are increasingly being met.
And would it be fair to say that the matter that we do find eventually function to them would rather support that they are there because they should be there, rather than there because of accidental viral infection?
Right. That would be my contention, is that they are deliberately there. And this also explains why you would see the same sequences shared, let's say, in humans and chimps. It could be that the share doesn't reflect common ancestry, but rather reflects a shared design, in the same way that, automobiles produced by different manufacturers have similar designs. It's because those designs work, right? Those are
yeah, sure. Go ahead. I think one of the major propositions or objections of evolutionists here would be that they would tell you, well, look, the locations of ERV insertions are even similar, and since reverse transcriptase, or the cut and paste enzyme, does not have, up to our knowledge, a location preference, then the similarity in the location of insertion of the ERVs can only be produced if common ancestry is true. I have some ideas of a response to that, but I would rather, of course, get your insights on it first. Yeah.
Yeah, and this is a situation where it really feels as if one hand doesn't know what the other hand is doing, because one of the things that I find intriguing is that for example, the J&J vaccine, the AstraZeneca vaccine, are adenoviral vector vaccines where people are using disabled viruses or inactivated viruses to carry, in this case it would be the gene for the SARS-2 coronavirus spike protein into cells, so that gene can then be expressed to produce the spike protein, which creates immunity. But what's intriguing is that researchers are utilizing the very properties of viruses that we consider to make them insidious to actually function as a gene delivery mechanism. And the technology that's being used, again, in J&J and AstraZeneca vaccines, it involves the adenovirus. But this is also a viral vector that's being considered in gene therapy, where people are looking to replace defective genes with healthy versions of genes. Or for those that are using CRISPR gene editing, these viral vectors are used to deliver the CRISPR gene editing package to cells. And so because of that, people study very extensively the insertion mechanisms, right? Because if you're trying to do gene therapy and you're trying to insert a healthy version of the gene into the genome of the patient, you want to make sure that that insertion is happening under controlled circumstances, that it's not just randomly inserting, because you may correct a genetic defect, but you're introducing another one through the delivery of the healthy gene, right? And so people study the insertion mechanisms, and what they've discovered is that they're actually hotspots, that these insertions are not always random. In fact, they are predictable where they're going to go. And now the implications of this are, let's say, even for the sake of argument, that some of the retroviral sequences are actually due to insertion events, right? These are not intentionally designed, but they're due to insertion events. Just because they're inserted in the same location doesn't necessarily mean that this is, again, due to common descent. It could be that these were independently inserted in the same locations in genomes because of this hotspot effect. That it's non-random, but it's highly predicted because of the nature of the sequences and the nature of the biochemical mechanisms at play. And in fact, this idea of insertion events being non-random has led some evolutionary biologists to argue that you can't necessarily use these sequences to build evolutionary trees, because it may not reflect shared ancestry, but independent events that took place in these different, in these different lineages. And so, but again, we're just beginning to develop an infancy. Our understanding is in its infancy of these kind of mechanisms. And what's interesting to me is that as these mechanisms, as we learn about the mechanisms and the pervasiveness of these mechanisms, suddenly we're seeing insights that are much more compatible with a design framework than with an evolutionary framework. Because again, the assumption when you see shared sequences is that the mechanism that produced that is random and it's a rare event. But if those events are not rare and they're non-random, you can't say it's common ancestry. You can't draw that conclusion, whatsoever. We're seeing examples of horizontal gene transfer, not only in microorganisms, that horizontal gene transfer where microorganisms can swap genetic material back and forth, creates a nightmare for evolutionary biologists trying to create evolutionary trees, as you can imagine, right?
Yeah.
Because different genes are going to give you different trees because, well, we're now seeing mechanisms that are supporting horizontal gene transfer in plants, where plants are swapping genes with one another. And that gene swapping is mediated by basically vectors, insect vectors that are infected with parasites that can then pick up and deliver genes to cells in those
Wow.
multicellular organisms. We're seeing
So
examples.
insects are not only cross-pollinating; they are actually even cross moving vectors, pieces of DNA from one plant to the other through parasites on the insects.
Yes,
It's
yes,
very
yes,
so complicated.
and we're seeing we're seeing this also with animals. And in fact, there's I can't remember the class of non-coding or junk DNA sequences, but you actually there's junk DNA sequences that are shared between cows and snakes, a particular species of snake and a particular species of cattle. And the
Wow.
assumption is that those shared sequences were essentially introduced from one genome into the other through some kind of horizontal gene transfer mechanism that probably involved, again, some kind of vector that
Wow.
and a parasite host or parasite that was parasitizing the vector. Yeah.
Wow.
So the point is that uh, we're still in our infancy in terms of understanding these mechanisms. We don't know how pervasive they are, but if these types of processes are pervasive, now it allows us to build a design model for genomes where we could say either the sequences have been intentionally designed by a creator, or some of those sequences may actually reflect mechanisms that are non-random and are highly reproducible mechanisms. Both of those could then, from a design perspective, explain,
Yeah.
the structure and the function of genomes, where you could then begin to say, well, if there's these differences from organism to organism, those differences may reflect an intentional design feature to create uniqueness, or they may actually reflect events that took place in that organism's genome, that were, mutational events after that organism was created. And so you could explain every feature in the genomes of organisms, and even explain comparative genomics from a design framework that doesn't rely on common ancestry.
Yeah. Well, I actually also have three additional. I think your argument is very insightful, actually. So thank you very much for that. I have three arguments that I would like also to know your insight about them. So the first one is, I'm not very sure what it means that an insertion is in the same location between humans and chimps, really. Because essentially, according to my reading, I don't think there is a single chromosome that is identical between human and chimp anyway. So if this is in location number, I don't know, 1000 on gene number 2 in humans, it cannot be in location number 1000 there. And the lengths of the chromosomes is different and everything. And at the end of the day, the comparison is based on synteny. At the end of the day, they just try to align the genes.
Right.
So I don't even know what it means that the insertion is in the same place. If it means that the insertion happens to be in, like after 10 genes here and 10 genes there is this insertion. But even if they are 11 or 12, then it is after that other gene in the human and that homologous other gene in chimp, then at the end of the day, the whole thing is a game of words, rather not actually same insertion location. And then in this case, what you have just said about hotspots would be a much logical explanation that for some reason the genome prefers that an insertion happens between this kind of gene and that next one, because when it's here, it has a specific advantage. If at all, this is an insertion, because
Right.
we are just seeing after the fact and trying to, in
Right.
hindsight, say that this must have been due to ancestry, as you say. So it's like as if we're spoon feeding the theory what we want. So this is my first. Let me spill out all the three, and then
Sure.
because they're sort of related. So this is my first contention, and then the second one is maybe combining what you have said and what I've just said. Maybe, for example, either the human line or the chimp line was infected by a virus. And then, like what we have seen in our age, that certain viruses jump from one species to another species, God knows why, and what triggers that, if not people playing with biochemistry, what naturally causes it, let's say. Because I don't think there was, bioweapon labs millions of years ago. But let's say that if at all humans were there millions of years ago, I don't think so. But let's say either there is a transposable element that is there in the design of chimp or in the design of human, or a viral infection happened in chimp or in human, and then there is this horizontal gene transfer mechanism, whether it moves from the human to the chimp or the other way around. And then the hotspot selection thing will cause it, when it moves from this species to the other species, go to the same place. So I mean, at the end of the day, it might not be at all a viral infection. It might be just a piece of the human or the chimp DNA has crossed over due to whatever reason. And then the third thing that I'm thinking, and I really want your insight about that, which also relates to what you have said about hotspots, but the hotspots thing is making it even much more crystallized. Correct me if I am wrong. I have never come across something that explains why certain genes are sitting on certain chromosomes anyway. So tell me if I'm wrong, but I have not come across something that says, for example, it is mandatory that genes related to sex are sitting on an X or a Y chromosome. But the matter of the fact is that they do. For some reason, genes that have functional relationship or are of a certain category happen to sit on the same chromosome or nearby chromosomes for some reason. However, it doesn't seem that we understand why that happens, or it doesn't seem that we have enough knowledge about what makes certain genes sit on certain chromosomes anyway. So, however, the observation says that genes that have functional relationship do sit on similar or on the same chromosome. And then if we can hypothesize from this that gene location in terms of which chromosome they are on relates to function, then maybe even the arrangement of genes, which gene comes after what other gene, also has significance. And then we can infer, or we can also hypothesize, that this makes certain places being good places for insertion also purposeful, except that we don't know enough about the whole thing that it is actually unfair to say, because it is the same relative location, even not the exact location, according to what I was saying in the first point, then this is the result of common ancestry. What do you think of this?
Yeah, I mean, I would, in effect, be highly sympathetic to all 3 points that you're making. And in terms of the second point you're making, this discovery of horizontal gene transfer in plants and animals, I think helps to supply, a mechanism to, to your proposal. So, and then the third point you're making to me is, I think, a really important point, because, this idea of the arrangement of genes on chromosomes, like synteny is the, the arrangement of genes on a single chromosome. There are some discoveries that are suggesting that synteny may actually have functional significance.
Oh, really?
Right?
Okay.
Yes.
Mm-hmm.
And so we're just making assumptions, that it doesn't, but, and it may not be that it's always functionally significant, but there are instances that I've seen published in the literature where it does have functional significance, right? And we are, again, in our infancy in understanding, the structure and the function of genomes. And if anything that the last decade or so has taught me is that, humility is probably a really good posture to have, and curiosity and wonder, because, this is where I think the design hypothesis actually can produce some very interesting science, because there's some very specific predictions that the design hypothesis makes about the structure and the function of genomes, where we expect there to be functional utility. That and even in these very early days, when people begin to approach genomes with the idea, could there be functional reasons for why things are this way, they are rewarded, for
Absolutely.
asking that question, right? As opposed to just saying, this is the way that it is. Let's not even bother with it. It's really interesting. I've been reading a little bit about Craig Venter, about, his life and his career. He's a fascinating scientist, that is one of the pioneers in genomics, and he began his career at the National Institutes of Health, and he was originally working on the Human Genome Project. And Craig Venter is a very impatient scientist. He wants, he wants to just cut through, all the clutter and do that last experiment first. And so he argued that the idea of sequencing the human genome, this would have been in the 1990s, I believe. He argued that the early 1990s, the idea of sequencing the human genome is a waste of time, because we know most of it is junk. So let's just sequence those regions of the genome that are expressed. And so he developed this technique where you would look at what are called express sequence tags. And in a few months, he had discovered a huge number of genes, looking at gene expression patterns in brain tissue. He discovered genes that nobody even knew existed in the human genome.
Wow.
And so this was emblematic of his approach. But thankfully, the Human Genome Project didn't take his advice. He got upset because he felt the Human Genome Project was wasting time. And so he left NIH and, got involved in other organizations, and, again, has played a very important role in the science of genomics, in part because of his impatience and his maverick attitude and things like that. He's a fascinating individual. But I think there's a lesson to be learned in that if we followed Craig Venter's approach, we would have identified, the genes eventually that are part of the human genome, but we would have been completely oblivious about the rest of the genome. And thankfully, the public project and then later Venter's work with Celera Genomics actually pursued the sequence of the entire genome. And through the use of the ENCODE project, we're now discovering, as we search for functional elements, that lo and behold, there's function in places that we would never have anticipated, because of an evolutionary mindset. But this is where I think the evolutionary mindset can be very dangerous to science, is that it shapes our perspective of biological systems as being cobbled together through evolution, as having no real reason, other than this is what an unguided process assembled, that if at first glance things don't make sense, we just deem it to be the non-functional vestige of an evolutionary history, and then we move on to the next problem. Whereas a design perspective would say, we may not know why it's this way, but we're confident that there's a reason for it. And so let's continue to study the system. Let's push on the system. And that approach is proving to be the more effective approach. People are discovering function in so-called junk DNA, not because they're intentionally looking for it. It's just as a happenstance because they're working on other problems, and lo and behold, they discover, to their surprise, function. But now there's enough of those types of discoveries that have accrued that people are really changing a mindset. But the mindset isn't changed because of the paradigm. The mindset is being changed because this is what the discoveries are pointing to,
because of
whether
compelling
we like
facts.
it or not.
Yeah. Actually, definitely great points. And I'll tell you why I'm proposing those 3 points that I told you. It is because from a software perspective, let's say you're writing this big program, whatever the program does, program that plays chess or program that's an e-commerce program or something. What programmers typically do, if you have a subroutine or a function that you need to call, it doesn't really make a difference where in the code you're putting the function, as long as it has a function name and it has a clear start and end. If you just move the module to this place or that place, it will still work when you call it. But we don't do that as programmers. We never do that. Only very bad programmers do that. What we will typically do is that we will have, for example, subroutines or functions or methods that relate, for example, to log in this place, and then something that relates, to whatever back end in that place, and something that is application servers in this place, and then things that relate to the front end in that other place, and things that relate to, I don't know, messages in this other place, etc. So a good programmer will have subroutines or functions or methods that have functional resemblance in chunks, okay? And then if somebody's looking at that after the code has been compiled, he will not appreciate why it's like that, because he has not had access to the uncompiled code. But if he knew how the uncompiled code in the original language is written, it's typically written in libraries. So you typically have the core code that actually does the real job. And you have, for example, a library for login, a library for security functions, a library for, rendering things on the screen, a library for controlling at whatever the screen and the mouse. And you have libraries, and you typically would link libraries by calling the whole library or DLL or whatever while you're compiling the code. So there are very obvious programmatical and practice-related and good programming sense-related stuff that will compel a good programmer to put pieces of the code that have functional relationships together. And you would only know that not if you are looking at the assembler code at the zeros of the ones that are the final program, but you will know that if you are somebody who actually does write programs. And since we don't write programs for creatures, thank God, not yet, to the best of our knowledge, and if we are doing, then that will explain plenty of nasty stuff that's happening. But since we're not, for example, creating, I don't know, squirrels from scratch or something yet, so we don't really know what it takes to create a creature. So we're completely oblivious of that. But for example, from what I see, there is an obvious wisdom for having, genes that relate to sex on an X or a Y chromosome, because then somebody can be a man or a woman by having the proper, whether XX or XY kind of combination. If it was all over the place, then we would have had very, strange kinds of human beings that are, not this or that. And the beauty of that is that you can make a distinction whether this is a male or a female by external shape and by voice and by the facial bone structures and by skeletal structure and by so many things. And the reason is that it's either XX or XY. If it was on the library of sex-related features was all over the place, what a mess that would have been, right?
Mm-hmm. Yeah.
So I think my guess is that the grouping of genes on chromosomes is definitely intentional because, as we have discussed before, the image of God thing has substance to it. And we have not looked into genetics while we were learning software, but we did the same thing. We arranged things in a certain way so that if we want, for example, that thing to run on Android versus Apple, for example, we would link the library that compiles it to that or that other platform. We don't just pick up 1,000 pieces of software from very different files. We don't do that.
Mm-hmm.
And we see that in application, actually. And then guess what? Even in the same library, typically you would arrange subroutines in a certain way. You start by a constructor method and end maybe by the destructor method and then other methods in a certain good practice or good sense way. And even, for example, on storage, you would, for example, put the directory structure first on the disk and the boot record first, and then the directory structure, and then storage space, and then you would have tapping or tapper that somewhere, like you have telomeres and centromeres, etc. Even these kinds of things we do when we arrange storage, etc. And then when we are writing a virus to infect a computer, okay, the virus would look for the boot record. It knows where the boot record is. And if this is an antivirus, it knows what places the virus will typically attack, whether it's attacking a program or attacking in storage space. So the arrangement of things, while completely arbitrary, we could have chosen the boot record to be anywhere. But we have those protocols so that we can work. And I think that this is actually what's happening in the genetic system, but we just don't know. So I think that all the arguments from ERVs and, that junk DNA and so do this and so do that, and is essentially a matter of ignorance that is actually completely ignoring our best practices in software development.
Yeah.
Because there are some people that would say, who said that this is programs? Who said that this is information? What is the definition of code? This is comply with the definition of language and this kind of stuff. What are your thoughts about this?
No, I think that what you're pointing out is that there is a rationale and a logic that undergirds how software is written, how software is essentially presented, and that that it's perfectly reasonable to think that the same thing would be true when we start looking at genomes, right? Is that and that we're already getting some hints that your ideas have real validity. But what I like is again, this is an example of science in action, right? Where we're taking our insights about how we design things and applying those insights to again, in this case, to genomic systems, with the expectation that our insights, in terms of how things are designed, will provide explanatory power. I suspect that the real insight into how genomes are going to be working is going to come from systems engineering at the end of the day, right? I think what's also going to happen too is that as we learn how genomes function and we think about them from a systems engineering standpoint, we're going to discover techniques that are being used in genomics that are going to propel systems engineering forward, that it's going to inspire new approaches to systems engineering. And so this is again another example that kind of intersects or overlaps with your ideas, that suggests that, again, design is really a fertile area of science, and is probably going to be instrumental in explaining, how genomes are structured and how they function.
All right, so I hope your predictions would happen soon in our lifetime, as fast as things are going already. So so thank you definitely for your comments. We have here a question from Omar who says, "How do ribosomes know what part of the DNA to use and when?" I think he means ribosomes. Then I think he would refer to RNA, or maybe he's like RNA polymerase instead of ribosomes, and then that would be DNA. So I think we'll be talking about that later when we're talking about transcription and translation. But if you can also give a brief answer, that would be great.
Yeah, there's essentially sequences in messenger RNA that will align with ribosomal RNA to position the RNA precisely in the ribosome so that it can begin the process of translation can begin. The shined Dalgarno sequences, what they're called.
So it's like the boot record in the case of the disk. That you have to always find or the padding sequences in the beginning of packets of communication that says the message is starting here, the message is ending here, kind of thing. It's amazing. It's again one more similarity between how we design communication systems and storage systems and programs. It's really amazing. We have here another comment, let's say, not question, from Utopia Buster, but I'm not sure why would you like to bust Utopia. Anyway, so he says the thinking cell for thought, memory, and action. So I asked him to elaborate, but I don't think he read my message. But what does this bring to your mind?
Well, this is actually something that biologists have noted about certain cells, particularly eukaryotic cells that are protozoans, single-celled organisms, is that they almost function as if they are being intentional about what they're doing. It's almost as if they're functioning like they have some kind of molecular level nervous system. Mm-hmm. And that's not the case, but they are behaving in that type of way. And we're going to be talking in a future episode about biochemical finite machines, or finite state machines. And so in other words, you can build algorithms, you can build, essentially computational systems that take input from the environment, and depending upon the state of the system and the input that's coming in, that system is going to respond in a particular way. And it gives the impression that there is memory, that there is forethought, that there is thinking going on, but in fact it isn't. At least not in the system specifically. It's just designed to behave that way. But the forethought, the memory, the design is actually in the mind of the engineer, the designer that produced that system. And so those features that you're seeing in cells is another aspect of cell biology that I think suggests creation, or a creator is responsible.
Yeah, it's just an example popped into my mind while you were talking. So I was thinking, for example, in a hot country, and you're buying a car that has an automatic braking system when there is ice, for example, and then it never snows in your country, and then one day it snows and you're driving fast, and then guess what? The system kicks in. Wow, my car has, foresaw that it would it would snow someday, and it just evolved this mechanism to automatically brake when it's skidding over snow. And actually, the car knows nothing except that the designer of the car, knows the dynamics of what it means to be, skidding over ice or
Right.
sliding over ice, and he has put that mechanism in the braking system or something to prevent skidding or sliding or so totally. It is strange to me, however, that evolutionary biologists will use a thinking cell, so they, at the end of the day, they know that the cell doesn't have a brain and doesn't have a nervous system or whatever, but they would refuse to say that it is the thinking God or the wisdom of God, or the wisdom of the designer is the one who's thinking, and the design is just reflecting his thinking.
Yeah.
So it's a philosophical standpoint. Here is from Tony Maurice. He says, Dr. Rana, are you familiar with the field work with abiogenesis by David Deemer? I think he buys nucleotides in high concentrations, and it has no relevance to prebiotic environments.
Yeah. I'm very familiar with David Deemer. In fact, I've interacted with him in the past, and in my book Origins of Life, and in Creating Life in the Lab, as well as The Cell's Design, I talk a little bit about some of the work that David Deemer has done, particularly with respect to the origin of cell membrane systems. But what Tony Maurice is bringing up is the idea that David Deemer is doing these prebiotic simulation studies and is seeing in the laboratory some intriguing results that suggest that maybe chemical evolution could explain certain steps in the origin of life. But what he's done is he's rigged the system in such a way to ensure a successful outcome. And one of the ways you can rig the prebiotic simulation experiments for success is by elevating the concentrations to very high levels. That ensures a greater likelihood of reactivity among the reaction constituents. But the problem is, on the prebiotic earth, you probably are looking at extremely dilute environments if you're dealing with either a local, pond or the ocean itself. But there are some mechanisms that origin of life researchers have explored to sequester materials and elevate their concentration. One of them is the use of clays. Clays have these layers, and the interstitial layers can accumulate organic material. So some people think that maybe clays could create these localized elevated concentrations. Others have argued, well, what if life arose under very cold conditions where you had ice on the surface of the earth as opposed to liquid water, and you have these interstitial phases in ice where through a process called zone refining, contaminants would accumulate in those liquid interstitial regions, creating very high elevated concentrations. But those scenarios have, we won't get into the details because of time, but both of those scenarios have other problems connected with them. So you might solve the problem of the need for very high concentrations for the chemistry to work, but you've introduced other problems that undermine, a chemical evolutionary explanation.
Yeah, he's also putting another comment. He's saying that Dr. Rana, vol clay, I'm not sure what vol clay is. It's a kind of clay, I think, is chemically stabilized and wouldn't be found on the early Earth. This is what
Yes.
I read from Dr. Tor. He said, yeah.
That's exactly right. And I talk about that point too in my book, Creating Life in the Lab, is the experiments that have been done where clay has been used to assemble RNA polymers only works when you're purchasing the clay from a particular supplier. So it's not just any clay, it has to be a particular type of clay. And then you have to treat the clay. You have to natriate it, meaning you have to remove all the ionic materials from it and replace them with sodium. And it's only that natriated clay that will actually work as a catalyst. So yeah, I would, concur with Jim Tor on that point.
Yeah. So one thing that I would like to ask you, and maybe we conclude with that, so we keep it below 1.5 hours. When I just look at all those scenarios that are drawn by, so Tony is having a closing comment. He says, atheist community and origin of life researchers are in total denial. God bless you both. God bless you, my friend. I just have this question. Let's assume we bring a set of bacteria or something, even unicellular eukaryotes or whatever, okay, and put them in a jar or something, and we dehydrate them so they are dead, okay. How much time? So we know they're dead, okay. So we kill them all, but we just dehydrate them, for example. So we preserve everything, all the chemicals and everything, so and the structures and everything, and then we add water, okay. I'm not sure if somebody has even done this before. How much time would it take for them to come back to life?
There are some bacteria that can survive for long periods of time under conditions of dehydration, but they are designed in such a way to do that. In other words, they've got mechanisms in place that allow them, under conditions of low water, to go into a state of suspended animation, where they are making use of low molecular weight materials to help protect the critical biomolecules,
Yeah,
so that
but
when
then it,
water is
yeah,
reintroduced, they can hydrate. So the microorganisms can do this, but it's not endemic in the molecules itself. There's a very elaborate system that they have in place to help them withstand dehydrating conditions.
yeah, but then it will not fulfill the condition that I have put, that we know that they're dead. So let's take a
Yes.
species
Yeah.
that doesn't have that feature, which seems to be a
Right.
pretty complex feature with plenty of underlying mechanisms, and it can sustain a sleep mood or a hibernate mood, kind of.
Right.
There's even some multicellular small stuff like what are they called, tardigrades or something.
Yes. Right. Yeah.
Yeah, they're called what, bears or something.
Yeah.
They have this funny name for them. That if you nuke them, they wouldn't die. Or they're very resilient. But let's not get those freaks in the formula. Let's assume that we're just getting bacteria or eukaryotes, unicellular species, and we just kill them, but we don't remove the material. We just remove the water, okay? So we know they're dead, okay? And we just then add water, okay? Will they come back to life if all the material is there?
Yeah. Probably. Uh,
Or let's
I,
say, let's even say, yeah, let's even
yeah.
say we get a set of proteins. We analyze the proteome, for example, of a specific organism or a set of organisms, okay, unicellular ones, and all the DNA sequences and all the lipids, all the building blocks that they need in their final form, and we just put them in a jar and add water and just. Will we come back after some time and find cells really swimming there?
That's an interesting question. I'm of the opinion that hypothetically it could happen. In other words, I see cells as being thoroughly chemical systems, where if you get the right molecules at the right levels and they're in an environment where they can interact in the right way, the processes of life are going to kick in, right? But so I don't. In other words, I don't see some kind of life force or anything like that. I think it's all explicable through chemistry and physics. But it doesn't. but to get the right chemicals at the right levels, with the right interactions is such a impossibility unless there are active mechanisms in place that are ensuring that actually happens. So in other words, so there's work being done in synthetic biology where people are trying to create protocells, and so in effect they're doing something similar to what you're describing in your experiment, where they're taking biochemical systems that they've purified and they're trying to reconstitute them into protocells that assume certain properties of life. But even just that process of taking parts and trying to reconstitute them is extremely difficult to do. And you have to have just exacting sets of conditions. You've got to have, highly skilled researchers using very expensive, chemical instrumentation to even take the first step towards producing something that is laughable compared to the complexity of the cell. So I think in principle it could happen, but what the work in synthetic biology is telling me is that even to reconstitute a system that was once alive requires an elaborate strategy and requires active intelligent input to even get the reconstitution to work. I don't know if that makes any sense.
Actually it does, and I'm glad that you gave this answer, because then the question is, now I think somebody must have tried it, because origin of life researchers are dying for creating life,
Right.
So that's why I asked the first question, and then I took it one level down. So your argument is, if we bring everything and we proportion everything, nothing in science would prevent it to come alive, because.
Yes.
But then my question is, let's take it one step forward and try to make it even easier. So everything is there because it used to be alive. I will bring 1,000, amoeba and 1,000 parameciums, and I'll just bring those guys there, and some ciliated bacteria, and whatever. Okay, so some fungi and some yeast, and put them, and I'll just kill them. But if I'll kill them by dehydration, so that all the good chemicals are there, all the stuff is there, and then the dehydration will be, the water will evaporate or something, and it's a closed system. And after I'm sure they're dead, I'll just re-inject the water with exact mass balance. Nothing will escape the system. And then the question is, why are they not coming alive?
Yeah,
this would be the question.
Yeah, I mean,
So,
my intuition
yeah.
is my intuition is that they would. My intuition is that they wouldn't. Nothing that was in that mix would come alive. That would be my intuition. I would be surprised if something did, right? And then if it did, I would say, well, there must be some unknown mechanism that's allowing this thing to be in suspended animation, though we think it's dead, right? So that would be my intuition, my expectation. And it's a good question because all the materials are there, but there's no active mechanism, I think, in place to get the materials in the right proportion and promoting the right interactions to kickstart the process of life.
Yeah.
And so it's not just simply the materials, but there's other input that has to go into the system to get it to kickstart the process, to jumpstart the process.
Absolutely. So I'm just trying to preempt the question of the origin of life.
Right.
Guys, you are putting so much effort into trying to figure out how prebiotic Earth could have produced the building blocks, and then how the building blocks came together, and then how they, formed organelles or membranes or whatever. Why don't we just cut through the chase, bring complete cells, kill them, re-inject the water, and just sit there for whatever time that you would like? And guess what? I have the same intuition like you, and I'm sure somebody has tried it. It couldn't have been like tens or maybe 100 years or something, or 150 years since Darwin. Nobody has tried it. Somebody must have tried it. And we've never seen even an animal that is dead, and we know is dead and came back. Okay, but let's say that bigger creatures have all those complexities and nervous systems and damage to brains or whatever. So I'm just going to the creatures that everybody knows does not have a brain and does not have this.
Right.
It's just chemicals. Okay. And since it does not come back to life, then life must have something beyond chemistry, whether this something is like an amazingly complex arrangement that relates also not just to adding water, but to adding water in a way that if you remove it, it will not come back to the same whatever crystal structure, van der Waals relations, whatever, but that is unhuman to do. And then it's game over. Let's just say that it's not a matter of just sticking chemicals together. It has to be a level of divine wisdom that can put them together in the right way, in the right time, in the right proportion. Even if you have all the proportions and all the masses and all the molecules, you cannot do it. It just doesn't that just end the question?
It should, right? I mean, what you're describing is doing the, what we used to call, when I worked for a Fortune 500 company, do the last experiment first, right?
Yeah, absolutely.
Right. We don't have time or money for you to develop an elaborate theoretical and experimental framework either. What's that one experiment we can do to decide if this is productive or not, right?
Absolutely.
And so yeah, I've never seen anything published where people describe that experiment and maybe somebody has done it. It would be interesting to see if that could be searched in the literature. But because then what you're saying is we're not assuming chemical evolution is needed to even produce the parts. We've got all the parts, right?
Absolutely. And it actually brings to mind the Frankenstein story, which is essentially more or less the same thing, like bring parts of humans that were alive, try to put them together, and guess what? No matter how many times you strike it with lightning or with electricity, this guy is not gonna walk. He's not gonna walk. And just make it simple. Bring the cells. Guess what? They're not gonna wake up, and we call it a day. And I think we'll leave people with that thought. Yeah.
You brought up Frankenstein, and I don't know. Have you ever seen the movie, the Mel Brooks movie, Young Frankenstein?
Young one? No, I only saw the adult Frankenstein.
Okay, well, Mel Brooks is would produce. He's not very active now, but he had a series of movies that were just spoofs, right? And one of them was a horror comedy called Young Frankenstein, and it's about Frankenstein's grandson who's trying to repeat his father's experiment, and he's got an assistant named Igor, and he sends him off to get a brain, and so he drops the brain he's supposed to get, and so he just grabs one off the shelf, and so things go awry because the monster is animated, and he's not very bright, and so Frankenstein asks his assistant, "So whose brain did you give me?" Well, I think it was somebody named Abby Normal.
Too bad for the Frankenstein.
Yeah.
Yeah. All right. So, so I think, I think the question is just like this: if it can't work for Frankenstein or the young Frankenstein, can't work for the cells, then we would just really need to call it a day. And here we have Man's Man. He says, "Salam everyone, just woke up and managed to catch the stream still in progress. How are you both?" I'm fine. How are you?
Yeah, good to hear from you. It's you're I think you're our favorite commenter.
Absolutely. I think this was a fun episode. Guys, I would also promise you that we'll go back to the topic of ERVs and pseudogenes and junk DNA, because we are going to discuss pseudogenes by the end of this first tranche of our series. Also, I have some news for you, because today's episode and the last episode, I had some extra editing to them, and I did not have the opportunity to add the editing to the premieres of the sessions. So I'll be probably posting them over the weekend with some extra illustrations and fun animations. So look out for that over Saturday and Sunday, inshallah. So Fuz, this has been a great, relaxed and fun discussion,
Yes.
full of plenty of insights, and I think it leaves some people here to think about things. And God bless you, and have a great weekend and a great time with family.
Yes. God bless you too.
Thank you. And for everybody, God bless you all, and have some great time tonight and over the weekend. And don't forget to share this with loved ones, and to join us next Thursday for our second installment about the Central Dogma, where next time we'll be talking about DNA replication, which is really a super topic and a very fun topic to learn about. So see you all soon. Assalamu alaikum wa rahmatullah. Peace be upon you. Thank you, Fuz.